All right, everybody, here we are, chapter 36. This is going to be resource acquisition, movement, and water potential. This one's a doozy, so we're just going to get right into it. So we're going to start by talking about water movement. Actually, the first thing I want to do is talk about this tree here that I have drawn. So I want to just talk about the general sort of resource acquisition that happens. So up here in the leaves of the tree, we're going to get light going in because we need that for photosynthesis, right? We are also going to be able to absorb atmospheric CO2 and send that back out as O2, right? And then out of the leaves, out of these stomata, remember stomata, we'll talk more about those later, we can lose H2O as well. So that's the four things in the leaves, and then down here below the ground, we can actually take in O2, and that's going to be for cellular respiration, right? And then remember, plants also are going to be carrying out cellular respiration and photosynthesis. Cellular respiration is the same for them as it is for us, so that's going to expel CO2. We can also absorb water from the soil and minerals or nutrients. So that's the big picture. So now let's break that down. First thing we're going to talk about for most of this chapter is going to be water movement. There will be a little bit of math. I'm so sorry. Water movement, remember water is transported through xylem. And water is going to go from high water potential to low water potential, which 99% of the time means it's going from roots to shoots. So I just used the word water potential. What is water potential? Think of water potential as a measure of how badly water wants to flow out of a system. So it technically is the measure of water's potential energy. I think of that as the ability of that water to flow. You can imagine if I take a balloon and I fill it really full with water and I tie it off. That water wants to leave. Like if I really filled it up good, as soon as I untie that balloon, that water's going to want to shoot out. It's going to want to flow. That means it has a high water potential. So that's one thing about water movement. We're going to go way more in depth on that in a second. But the next sort of topic is going to be transpiration. So transpiration is sort of one of the driving forces that lets us take water from the roots and move it upwards against gravity. Transpiration starts with evaporation out of the leaf. So I want you to sort of imagine I'm going to track one singular xylem here. So one single tube of water. There it is. So the first thing that's going to happen in transpiration is it's going to go to a leaf. There's a leaf. That leaf is going to have open stomata. And water molecules that are here are going to evaporate out into the atmosphere. The process of those water molecules leaving is actually going to pull other water molecules upward closer to that area. And that is going to be a combination of pressure and capillary action, which we're going to break both of those down very in depth. So that's transpiration, is the evaporation pulling water upward. So capillary action refers to sort of a combination of some of the properties of water, cohesion and adhesion. And what it looks like sort of visualizes here. We have a tub of water, and I've put 3 tubes of different thickness in that water. The first tube is really wide, and so it just kind of sits in the water. You can think of these as straws. We'll say straws. The second one is a little bit thinner, and the water level is actually higher right here. Water has actually traveled up a little bit. And then in this really thin tube, water has traveled really far up. The reason it can do that is because the water molecules have more adhesion towards this super thin surface, it's easier for them to cling onto the side of that, right? So capillary action refers to cohesion and adhesion of water, helping it defy the force of gravity. How this relates to transpiration is, let's say we have a water molecule here that evaporates. When that water molecule leaves, it's going to do two things. One, it's going to pull other water molecules towards it because of cohesion, but sort of the bigger thing is it's going to leave behind an empty space. And when there's an empty space, that means pressure has decreased. You can think of it as if I have an empty water bottle and I suck on the water bottle. You know how the water bottle shrivels up and shrinks? That's because we've taken the pressure out of it. And that creates what's called a tension force. Tension is pulling. So you can kind of think of positive pressure as a push and negative pressure as a pull. So we have a negative pressure that pulls things upward. And I'm going to try to explain pressure better later. Well, actually right now. So the next thing, we're going to go back to water potential. Remember I said water potential is the ability of water to flow. How bad does water want to flow? A high water potential means it wants to flow really bad. It wants out of there. A really low water potential means that it doesn't want that water to leave. It might even want more water. And water will always move from high water potential to low water potential. So water potential is denoted by this symbol here. I don't know what it's called. It's definitely Greek, as many symbols are, but it looks kind of like a trident. You know, like Poseidon, he had a trident, he was god of the sea. So water potential, water. The water potential of a single like pool of water or a system is equal to the pressure potential, which is a trident with a little p, plus the solute potential. So the pressure potential is almost always a positive value. So if it's always a positive value, that means it's going to increase our total water potential. And if we're increasing our total water potential, that means that it's going to want water to flow more. And then solute potential is almost always negative. In fact, I think it is always negative. And that means it's going to lower our overall water potential, which means it's going to be less likely to want to flow. So there's sort of two opposing forces here. So let's break them down. We're going to start with solute potential. So again, solute potential is always negative, and it determines the movement of water via osmosis. You may remember osmosis from last term. If you don't, osmosis is the movement of H2O, movement of water through a semi-permeable membrane to balance concentration. So the best way to think of osmosis is if you have two solutions separated by a semi-permeable membrane. So here we have A and B, and all of these little dots in here are going to be solutes. So you can think of them as ions. Maybe we have salt water, but this water in B has a lot more salt than this water in A. But a semi-permeable membrane is put between these two. So these ions can't move back and forth at all. The only thing that can pass is water. So we are going to go through, and I'm going to start by saying, remember, solute potential is negative and lowers, means less likely to flow. So a higher concentration of ions is going to make the solute potential more negative. And if our solute potential is more negative, that means it's going to lower the overall water potential. So let's take a look here. So A and B, one of these is going to have a high solute potential, and by high I mean a very negative solute potential, and one will have a less negative solute potential. So which one's which? Well, I said a higher concentration is more negative. So that means which one of these is going to be more negative for the solute potential? It's going to be B. So I need to Google what symbol this is so I can't just keep saying like trident. It's called psi, PSI, like Poseidon, psi. So psi S, so the solute potential of B is going to be more negative. And the solute potential of A is going to be less negative, so closer to 0. So what effect are each of those going to have on the overall water potential of the solution? And for this instance, we're going to assume that pressure is the same. So we can eliminate this guy. Well, if this is less negative, that means we're taking away less from the water potential, right? Because remember, we're adding a negative, right? So we're essentially subtracting the solute potential. So a more negative solute potential lowered the overall water potential. So the one with the more negative solute potential, which is B, is going to have a lower overall potential. So this is going to be lower, which means this one will be higher. So if I know that solution A has a higher water potential than solution B, which way is water going to flow? Is water going to flow from A to B or B to A? Hopefully everyone is thinking A to B because that's correct. So water is going to pass this semi-permeable membrane, and you'll actually end up with something that looks like this. So no ions moved, water moved. This was osmosis. And our water went from high water potential to low water potential. And in this case, it went from the less negative, or the higher, technically the higher because it's closer to 0, solute potential to the more negative solute potential. All right, so that's one piece of the puzzle. That's solute potential, check. Next up is going to be pressure potential. So pressure potential is always going to be a positive value, which means the higher your pressure potential, the higher the magnitude of your pressure potential, the more water potential you will have. So as pressure potential increases and becomes more positive, water wants to flow more, and that's going to make a lot of sense. I want you to think of a water balloon that I put inside of a box. The water balloon is going to represent the cell, and the box is going to represent a cell wall. So at first I have this water balloon in the box, and I fill it with like some water, but not much. So I just put a little bit of water in. You see how it's not really pressing on the sides, it's not pressing on the box at all. So this doesn't have much pressure to it at all. You know, this little valve being open doesn't really matter. It doesn't want to flow. The water doesn't have a lot of pressure pushing it out. But then if I add water and I fill this balloon up more, you can see it's starting to press on the sides. So now that pressing on the sides is going to have a little bit of pressure to it. It's going to be sort of pushing water out a little bit. Water wants to leave a little bit more now. And then if I really fill this balloon up with water, you can see the sides of the box are actually starting to like bend out like just a little bit because I've filled that with so much water. So now there's a lot of pressure because that box wants to hold its rigid shape, but it's being forced to bow out because I've put so much water. So pressure is increasing as I add water to this system. And there are actually two pressures acting here. There's going to be one that is going to be my balloon pushing on the box. And that balloon pushing on the box is going to be referred to as turgor pressure. This is the one that we're going to use a lot in the next couple chapters. So the turgor pressure is the pressure of the balloon pushing on the box. But there is also a pressure because remember I said that the box wants to hold its shape. It wants to be rigid like this, but it's forced to bow out like this. So the wall, the box or the cell wall is also exerting a pressure inward on the balloon. Because I didn't say it, but that turgor pressure is the balloon pushing on the wall. So that's an outward pressure because we're pushing on the wall and the wall is that way in this little part here. But the wall is going to be pushing back because it wants to keep its form. And that's the wall pressure. And these pressures are actually equal and opposite. So the turgor pressure here is equal to this wall pressure. They're just acting in separate directions at equilibrium. That does shift a little bit, but turgor pressure is the one that we care more about. So we have these pressures acting. We can say as a rule, as pressure increases on the system, so the more I fill up this balloon, that's going to increase my pressure potential, which is psi p, or the trident with a p. And if we're increasing the pressure potential, we said that it's going to increase the water potential. So it's going to make water want to flow more. It's going to give more potential energy to that water. So this is the highest water potential of these three, and this is the lowest. So if we think of a water balloon again, now we're going to think of it as I have a water balloon and I fill one up with just like a tiny bit of water and then I tie it off. But there's another one that I fill up to the max and I have to like pinch it off and tie it off really carefully. It's about to burst. So we have this one that's just slightly full and I tie it off. And then I'm going to untie both of them. When I untie this water balloon that I didn't fill up really at all, I put maybe just a couple drops of water in, that water's not gonna it's not gonna squirt out at all because there's no pressure. There's no pressure acting on it to push out. But the one that was very, very full, as soon as I untie it, the pressure of the balloon wanting to go inward is gonna push that water out. It's gonna make that water flow. And that's gonna be a very high pressure potential, which means it's going to be a high water potential, and that's why water is flowing. And then this one is going to be a really low pressure potential and a really low overall water potential. So that's sort of the balloon analogy that I like. So now we're going to bring this together. So we know what all of these variables mean. So water potential is going to be measured in megapascals. Sometimes it's going to be in bars. If you need to convert those, Dr. Reddy will provide like the conversion factor for you, hopefully, or you can just answer it in either of these. And so bars and MPa, megapascals, are pressure units. So we're kind of measuring the pressure. And it is we are comparing two solutions usually in these kinds of problems. And remember, water is going to move from a high to a low pressure potential. So two sort of forms of this question might be like this. So we have this picture here with two solutions, all right? Solution 1 and solution 2 are separated by this semi-permeable membrane. So we can see that solution 1 has very few solutes and solution 2 has a lot of solutes. We can also see that solution 1 is sort of making the semi-permeable membrane bow out. And if that's a bowing out, I drew it like that to sort of indicate that there's pressure here. There's more pressure coming from this side than this side. So let's go through and think about the variables. So let's start with the pressure potential. So the pressure potential is going to be higher on which side? It's going to be higher on solution 1 because we have more volume. More volume is more weight, and so it's pressing more on this semi-permeable membrane. So there's more pressure over here and less over here. So we have a high pressure potential here, so more positive, and we have a less positive pressure potential here. Solute potential, we have way more concentration here than here. So the solute potential will be higher, more negative for solution 2, and it will be less negative for solution 1. So then we can determine which one has higher water potential. Because if this one had a high positive and a low negative, that means, just to put random numbers to it, let's say we had 60 and -5. that would mean the water potential would be 55. And then to put random numbers over here, let's say this had a negative pressure potential. Let's say this had a pressure potential of -5 and a solute potential of like -40. That's going to have a -45, and we'll say megapascals. So solution 1 has a higher water potential than solution 2, so water will flow from solution 1 to solution 2. Very likely, sort of the types of math questions you'll get she will just give you the pressure potential and the solute potential. So let's say for solution 1, we have a pressure potential of 8 bars and a solute potential of -3. And then for solution 2, we have a pressure potential of 6 bars and a solute potential of -4. How do we determine the flow direction? Well, we remember our equation that looked like this, and we figure it out for each one. So solution 1, we have solution 1 is going to equal the pressure potential of solution 1 plus the solute potential. So that's going to be 8 plus -3, and that's going to equal 5 bars. And then over here for solution 2, we have a pressure potential of 6, and we're going to add -4, which is going to equal 2 bars. So flow will happen from solution 1 to solution 2. One thing I want to talk about that might trip some people up is negative values. If I have 2 solutions, and solution 1 has a water potential of -30 bars, and solution 2 has a pressure potential of -32 bars, remember, high to low, don't get tripped up by negative signs, basically, is what I'm trying to say. -30 is higher than -32. It's more positive than -32. So this would also be solution 1 to solution 2. Just a quick side note, because I don't want anyone to get too tripped up on that. All right, next thing we got to talk about is types of solutions. So when you talk about solution types, you're going to talk about the tonicity of a solution. Tonicity, by definition, is a solution's ability to change the water volume of a cell in that solution via osmosis. So tonicity is a comparative term. This is really important. You can't just ever say that a solution is hypertonic. That doesn't make sense. It has to be hypertonic compared to a cell, or hypertonic to the cell. We'll get more into that in just a second. So it's always a comparative term. So there are 3 types of solutions. We have isotonic and isotonic. Remember, iso means same, so same tonicity. So that means that the solute concentration is the same in the solution and in the cell. So that means that if I have a cell full of water and I put it in a bucket full of water, and both, let's say that it's salt water. Let's say that there's salt water in the cell and there's salt water out of the cell. If it's the same concentration of salt in both of those systems, then they are isotonic to each other. Hypertonic means that there's a higher solute concentration in the solution compared to the cell. So that would be a hypertonic solution. So that means that the water that I put my cell in is really salty, and the water inside the cell is not very salty. Then the last one is hypotonic. Hypo means low, and that means a hypotonic solution would have a lower solute concentration in that solution compared to the water in the cell. So this would be, I take a cell that has a lot of salt inside the cell, and I put it in fresh water that doesn't have any. So again, on the comparative terms, things you could say. You could say, I have a cell, and that cell is in an isotonic, hypertonic, or hypotonic solution. So let's say that I said, oh, the cell is in a hypertonic solution. That means the cell is in a solution that has more concentration than my cell. You could also say that the cell is, you could just say, like in that same, like if we're using hypertonic, I could say the cell is hypertonic to the solution. Now, be careful here. If I say the cell is hypertonic to the solution, that means that the cell has more concentration than the solution. The other thing you could say is the solution is hypertonic to the cell. And that would mean that the solution is saltier than the inside of the cell. So a quick note, if a cell is hypertonic to the solution it's in, that means that the solution is hypotonic to the cell. So whenever you see these terms, read them carefully and read what is being compared to what. So here is sort of, I drew it out a little bit. So here I have an isotonic, hypertonic, and hypotonic solution drawn. So in this, so in each of these scenarios, I have a cell that I've drawn, and these cells all have the same starting concentration. And then I have differing concentrations of solution. So the concentration of all of these cells, and let's just say we're talking about salt, why not, is 3 molarity. Molarity is just is the unit of concentration. Don't worry too much about it. So in an isotonic solution, I have this 3 molarity concentration cell, and I'm putting it into an equal concentration solution. And so when I do that, no change is going to happen. And if no change is happening, so that means water is moving in and out at the same rate. There's no change in the volume of either one. And if that's the case, we know that the water potential of this cell is equal to the water potential of the solution, and the cell is going to remain in a flaccid state. So now let's talk about a cell that I put into a hypertonic solution. So the cell is going to be in a hypertonic solution. So if I'm putting a cell in a solution that is hypertonic to the cell, that means that the concentration of the solution is higher. So we still have our 3 molarity cell, and then we have a 6 molarity solution. So there's much more salt in this water than there is in the cell. So when I put that in, let's think about this in terms of solute potential. The more concentrated something is, the more negative its solute potential is going to be. And remember, the more negative the solute potential, that is actually going to lower our water potential. So more solute means that the water wants to leave less. So now I put my cell in. Now my cell has less concentration. My cell has a less negative solute potential. It's closer to 0. And that means that it has a overall higher water potential. So in this situation where I place it in a hypertonic solution, the water potential of the cell is going to be greater than the water potential of the solution because the solution is more concentrated, which means it has a more negative solute potential. So that means that water is going to flow out of the cell and into the solution. And that means that the cell is going to shrivel up and become tiny like that. And when that happens, it means that we call that the cell plasmolyzing. The cell is plasmolyzed. That means it shrivels up because water leaves the cell, because that water is going from the high water potential to the lower. And the last one is a cell that I'm going to put in a hypotonic solution. So we have our 3 molarity cell, in a 1 molarity solution. So now we have a more negative solute potential inside the cell and a less negative solute potential in the solution. So when I put this cell in here, what's going to happen? Well, which is higher, the water potential of the cell or the solution? The answer is the water potential of the solution is going to be higher because it has a closer to 0 or a less negative solute potential. So we have now the water potential of the cell is actually less than the water potential of the solution, which means water is going to go from the solution into the cell. And when that happens, it's going to swell the cell up, and the swell is almost, sometimes it does burst. And so H2O entering the cell is going to swell the cell up, and we call that a turgid cell. And this is for plant cells specifically because we have cell walls. So that cell is now turgid. It has a lot of pressure now. All right? So that's the 3 kinds of solutions. All right, so next we are going to talk, we're going to switch gears. We're done with math for now. Thank goodness, right? Get that out of here. Now we are going to talk about the roots of water movement in plants. And I'm talking about, we're going to first talk about like how it gets to the xylem. Because remember, we're going to absorb water through the roots, and it's going to go to the xylem where it can then go up the plant through that process of transpiration. Right? So, we are going to start by defining the 2 pathways. There are 2 pathways. One is called the apoplastic pathway, one is the symplastic. So apoplastic pathway moves water through the cell walls and intercellular spaces of plant cells. This method of transport is going to be fast, so that's a good thing. It's going to be passive, so we don't have to use any energy, but it's going to be unregulated. That's bad. When I say unregulated, I mean there's no filtration. So that's going to be a problem. But we'll have a solution for that. So this route starts in the soil, and then water is in the soil, and then water will be absorbed by the root hairs, and then it will go to the epidermis, and then it will go to the cortex, which, remember, the cortex is where our vascular tissue is, and it's going to get there by going around cells by using the cell walls. But this apoplastic pathway is going to have a blocker or a stop sign that is called the Casparian strip. So the Casparian strip is a band around the cell wall that's made of a lipid or a fatty substance called suberin. Remember that word, that could be a test question. So the Casparian strip is a band that forms around cell walls of suberin, and if it's a lipid, lipids are hydrophobic. So when the water hits the Casparian strip, it does not want to continue. And so I've illustrated the apoplastic pathway down here. So this is apoplastic. We have water entering and traveling through the cell walls. So it never goes in the cell until it hits the Casparian strip. And we have this band all the way around. Hits that Casparian strip, and then it's going to go into the cell. Because remember, the Casparian strip is only around the cell wall. So then it's going to force into the cell. And the route of water movement inside the cell, that's just this nice straight line, is called the symplastic pathway. And it's the cytoplasmic route. You can think of it as SYCY, cytoplasmic route. It passes directly through the cytoplasm of cells, really utilizing these plasmodesmata. Remember plasmodesmata? I talked about it last video very briefly. So these are plasmodesmata, which are kind of modified cell junctions. This process is going to be slower. It's not going to be able to move through the cytoplasm as fast, but it's going to be regulated. The cell can decide what moves in and out of the xylem this way. So it's going to be much better for regulation. And again, those plasmodesmata help it go right through, and it avoids the Casparian strip. I didn't say it, but that Casparian strip is very common in the endodermis, which, if you may remember, is the sort of the protective layer around vascular tissue. So we have our apoplastic pathway through the cell walls and our symplastic pathway through the cytoplasm. A couple more pages, we're almost there. So now we have water in our xylem, and remember the xylem is the tubes that are going to carry water upward. So now I want to talk about root pressure. So root pressure is the pressure that roots exert to push water upward through the xylem. And this happens for a couple of reasons. So root pressure, we have water down here in the roots. The other thing that roots absorb is ions. So as ions build up here and water builds up here, that means we have concentration. So we have, I'm just going to use salt because I always do anytime I talk about concentration. So if I have solute potential here, but in this xylem, there's no salt. There's no ions in this xylem. So that means the water potential in the roots is going to be higher than the water potential in the xylem. And that is going to generate a little bit of pressure. That's going to help generate this root pressure. So root pressure in part is a result of ion buildup in roots, which creates a more negative solute potential, which lowers the water potential of the root, meaning it wants to pull in water. Sorry, I said this wrong when I was describing it down here. This is pulling water in, this because we have concentration inside the roots. I'm sorry, guys. So this concentration inside the roots is going to cause water to want to enter the roots. And the buildup of water in the roots creates pressure, because the more we have a fixed space, it's kind of like, again, trying to overfill a water balloon. We have roots which have a definite volume, but we're jam-packing them with water, which is going to create pressure. And that happens because of the ion concentration, which, again, I'm going to say it again because I messed it up. The ion concentration is higher in the roots as ions build up, and that creates a more negative water potential in the roots, which draws water in, creating pressure. The other factor in root pressure is that transpiration that we briefly talked about. So transpiration, again, I have sort of a step-by-step process here of it. So the first thing that's going to happen is evaporation. Stomata on this leaf are going to open and water is going to leave. So this water molecule is going to evaporate. Once that water molecule is evaporated, a couple things happen. One is we now don't have anything here. There's just empty space here. And empty space creates negative pressure. So step one was evaporation. Step two is this negative pressure that we generate, which is called tension. And tension is a pulling force. So that's going to help pull this next water molecule up. The other thing that's going to help pull that water molecule up is cohesion. Cohesion means H2O attracting H2O as a result of hydrogen bonding. So this water molecule evaporating and leaving is going to pull this water molecule up even more. So we have tension and cohesion working with us. Part of the problem, though, is we are working against gravity. And one of the things that's going to help alleviate that is adhesion, which is water sticking to surfaces. So what this means, part of the reason that we can sort of not have this water fall back down is because it's also going to be attracted very weakly to the sides of the xylem, which is going to be really helpful for it to sort of resist a gravity. And all of these forces also kind of create this surface tension effect of water, which is further going to help it resist gravity. So this is root pressure. This is all root pressure, and this is also called the cohesion tension theory. So remember, evaporation is going to create negative pressure that we call tension, and that water molecule evaporating is also going to pull the next water molecules up with a little bit of force because of cohesion. They're going to stay there because of adhesion and surface tension. So that's root pressure, and that is the cohesion tension theory. Next up, this one's nice and easy. We're talking about stomata and water loss. So stomata, the singular of stomata is stoma. So a stoma will open and close because it has two guard cells around it. So if you really zoom in on a stomata, it kind of looks like this a little bit, and this is the actual opening here. So the water content of these two cells, and these are our guard cells, is going to determine if the stomata is open and closed. If these guard cells are chock full of water, they will change shape a little bit just due to like their own unique physiology, and they will open. So this is when these guard cells are full of water or turgid. If you remember, we had turgor pressure and we talked about a turgid cell. So turgid means full of water. So when they're full of water, stomata will open. So this is an open stoma. And when a stoma is open, that means that H2O can leave. So that means evaporation happens. And it also means we can take in CO2. Take in CO2 from the atmosphere. Alternatively, though, and this kind of makes sense if you think about it too, because if these cells are full of water, then that's kind of a signal that the plant has water. So when the plant has enough water, it can afford to lose some water through this evaporation. On the other side, though, we have when the guard cells are flaccid, then they're shaped more like this. And then we have a closed stoma, which means we are not losing water, but we're also not taking in CO2. 95% of water loss of a plant is through stomata opening. The reason you don't have it everywhere is because of that waxy cuticle that your dermal tissue can secrete. So there are also two adaptations I want to talk about that plants have to minimize water loss. So there's a group of plants called cam plants, and cam plants temporally separate systems, and that means they separate them using time. Cam plants will only have their stomata open at night, and then they'll just store the CO2 in some organic compounds that can then be used during the day for metabolism. So this is really good in areas that are very, very dry. And the reason this is good in areas that are very, very dry is by only opening stomata at night when, like, for example, the sun isn't out, evaporation won't happen as much, so we can stop some of that water loss. The other plant modification is C4 plants, which it's called C4 because of the compound that it will turn CO2 in. So in plants we learned about in photosynthesis, CO2 was fixed into a 3-carbon sugar, but this time it's going to do it in a 4-carbon sugar. And what this is going to do is reduce photorespiration, which is using oxygen instead of CO2 for the Calvin cycle, which is really inefficient energetically. It's really bad. But what this does, if we fix this into a 4-carbon sugar and we spatially separate the processes of carbon fixation, then it's going to allow our stomata to stay closed more because we don't need as much CO2, because we have plenty of CO2 stored in these 4-carbon compounds because we're not wasting stuff on photorespiration. So this is good in hot, moist areas that have plenty of water, but are also really hot and therefore sort of drying out. So this is good in like a tropical area. And again, this spatially separates CO2 fixation. So C4 is spatial, CAM is temporal. All right, we're at 45 minutes, but this is the last thing. We've been talking about water movement in xylem a lot. Now we're just going to talk about sugar movement, and it's pretty easy. So sugar movement moves from the source to the sink. So source meaning where we have sugar stored or being produced, sink being where we need sugar. So during the growing season, our source is going to be leaves and stems. Remember, leaves are going to do a lot of photosynthesis, which makes sugar. And the sink during the growing season will be meristems, which is growth regions or fruit, and sometimes roots as well. But in the early season, when sort of the plants are just developing, it will have stored all that sugar from the last growing season in the roots, storage tissues. And so the sink will be new leaves when it's trying to make new leaves so then it can do more photosynthesis. So phloem, remember phloem is the tubes that transport sugar, have two elements. They have sieve tubes and companion cells. So here we have sieve tubes. This is one sieve tube cell, and this is another sieve tube cell, and they're connected via these sieve plates. And these sieve tubes can receive water from the xylem as a passive process. So water can jump over to these tubes, and the tubes is where is the transport part, right? So, and then we're gonna have a source down here. So let's say that this, since we're underneath, we'll say this is the root, and we have stored sugar down here. So we have a whole bunch of sugar right here. To get this sugar into the phloem, we have to first get it to the companion cells. So the companion cells are going to build up a bunch of sugar, but then eventually we run into a problem where the concentration of sugar in the source cell is lower than the companion cell. So we have a high concentration here now and a lower concentration here. How do we get something to go? Because we don't need the sugar in the source, we need all of it to go. And I don't want backflow because I need things here. So this is going to have to require secondary transport. So to get sugars from the source to the companion cell, sometimes you need secondary transport. And if you remember from last term, secondary transport means we are going to indirectly use ATP. And in this case, the secondary transport is going to be the proton sucrose symporter. So here we have outside the companion cell and inside the companion cell. So to have secondary transport, we first have to have primary transport. And the primary transport in this case is going to be a proton pump. So proton pump is going to hydrolyze ATP to ADP, right? So we're going to cleave that phosphate, which generates energy, and we're going to use that energy to pump protons against its concentration gradient and get a very high concentration of protons outside of the companion cell. And then the secondary transport can happen because now we have, thanks to this proton pump, a very high concentration of protons, and we also have our sucrose out here. And remember, we want our sucrose to go from an area of low concentration to high concentration, and that is against its gradient. So what sucrose is going to do is hitch a ride with the protons as they come back in, because we have this high concentration of protons outside. So this symporter is going to let the protons back in, which is going to generate energy because these protons are now moving down their concentration gradient and generating some kinetic energy. Sucrose is going to hop in on that kinetic energy and go, yes, I'll use this to move against my concentration gradient and get me inside the companion cell. So that's how we get sucrose to build up in the companion cells So this was a symporter, and remember it's symporter because these protons in sucrose is going in the same direction. The other type of secondary transport is an antiporter. And an antiporter would be if we had them going in opposite directions. So let's say that we have our protons moving in here, but just like, let's pretend that that I want to get a sucrose from here to here, but sucrose is higher outside. So this would be an antiporter. So antiporter is just secondary transport where things are moving in different directions. Symport, same direction. And remember, all secondary transport is going to have one thing moving with its concentration gradient, which is the protons in this case, and one thing against its concentration gradient, which is sucrose in this case. So the last thing, we now have them in the companion cells, and then the companion cells can transport them into our sieve tube cells. And once that happens, it's going to build the turgor pressure in this tube cell, which is going to push them upward into this cell, where it can then go through the companion cell into the sink. And now we're just moving based off turgor pressure. So the turgor pressure at sources are higher than at sinks, so that's the way the sugar will flow. All right, that was a long one, but that's it for this chapter. Let me know if you have any questions, and have a good one.